Bismillahirrahmanirrahim. In this video, we're going to learn about different types of neurons and glial cells. Let's begin. The nervous tissue is composed of two types of cells, neurons and glial cells. Neurons are the primary cell type of the nervous system. They are responsible for communication and integration that the nervous system provides. They are electrically active and release chemical signals to target cells. Glial cells or glia are known to play a supporting role for the nervous tissue. Neurons are important, but without glial support, they would not be able to perform their function. Neurons are the building blocks of nervous tissue. They are responsible for the electrical signals that communicate information about sensations and that produce movements in response to those stimuli, along with inducing thought processes within the brain. The main part of a neuron is the cell body, which is also known as the soma. The cell body contains the nucleus and most of the major organelles. Cell body have many extensions which are generally referred to as processes. Neurons are usually described as having one axon, a fiber that emerges from the cell body and projects to target cells. That single axon can branch repeatedly, forming numerous axon terminals to communicate with many target cells. It is the axon that propagates the nerve impulse, which is communicated to one or more cells at specialized areas of contact called the synapses. The other processes of the neuron are dendrites, which receive information from other neurons at synapses. The dendrites are usually highly branched processes, providing locations for other neurons to communicate with the cell body. Information flows through a neuron from the dendrites across the cell body and down the axon. This gives the neuron a polarity, meaning that information flows in this one direction. Many axons are wrapped by an insulating myelin sheath, which is actually made from glial cells called Schwann cells. There are gaps in the myelin covering of an axon. Each gap is called a node of Ranvier and is important to the way that electrical signals travel down the axon. The length of the axon between each gap, which is wrapped in myelin, is referred to as an axon segment. Where the axon emerges from the cell body, there is a special region referred to as the axon hillock. This is a tapering of the cell body towards the axon fiber. From the axon hillock to the first Schwann cell, the bare area of the axon is referred to as the initial segment. The first way to classify neurons is by the number of processes attached to the cell body. Unipolar cells have only one process emerging from the cell. true unipolar cells are not found in humans. Human pseudo-unipolar cells have a process that emerges from the cell body, but it splits so that at one end are dendrites and at the other the axon. Pseudo-unipolar cells are exclusively sensory neurons and have two unique characteristics. First, their dendrites receive sensory information, sometimes directly from the stimulus itself. Secondly, the cell bodies of unipolar neurons are always found in ganglia. Bipolar cells have two processes which extend from each end of the cell body opposite to each other. One is the axon and one the dendrite. Bipolar cells are not very common. They are found mainly in the olfactory epithelium and in cellular layers of the retina. Multipolar neurons are all of the neurons that are not unipolar or bipolar. They have one axon and two or more dendrites, usually many more. In human body, most of the neurons are multipolar. Neurons can be classified on the basis of their function into three types. The sensory neurons are part of the afferent division of the nervous system. These transmit impulse from sensory receptors to the integrating centers in the spinal cord and the brain. The cell bodies of sensory neurons are located in the dorsal root ganglion of the spinal nerve. Motor neurons are part of the efferent division of the nervous system. These transmit impulse from control centers in the spinal cord and brain to the effector organs like muscles and glands of the body. The cell bodies of motor neurons are located in the ventral root ganglion of the spinal nerve. Interneurons are neurons that are found exclusively in the central nervous system. Interneurons act as middleman between sensory neuron and motor neurons. It also connects to other interneurons, allowing them to communicate with each other. The interneuron enable integrative and control function of the nervous system. Glial cells or neuroglia or simply glia are the second type of cell found in the nervous tissue. They are considered to be supporting cells and many functions are directed at helping neurons complete their function of communication. There are 6 types of glial cells. 4 of them are found in the central nervous system and 2 are found in the peripheral nervous system. The table seen here outlines some common characteristics and functions of the glial cells. Glial cells found in central nervous system include astrocytes, oligodendrocytes, microglia and ependymal cells. The satellite cells and Schwann cells are found in the peripheral nervous system. Astrocytes and satellite cells are believed to provide structural support to the neurons. The myelin sheath is formed by oligodendrocytes in central nervous system and by Schwann cells in the peripheral nervous system. Microglia function in immune mechanism and are believed to be phagocytic cells in the central nervous system. The ependymal cells form cerebrospinal fluid. Now let's look at some more details of individual glial cells. The astrocyte appears to be star-shaped under the microscope. Astrocytes have many processes extending from their main cell body. Those processes extend to interact with neurons, blood vessels, or the connective tissue covering the CNS that is called the pia mater. Generally, they are supporting cells for the neurons in the central nervous system. Some ways in which they support neurons in the central nervous system are by maintaining the concentration of chemicals in the extracellular space, removing excess signaling molecules, reacting to tissue damage, and contributing to the blood-brain barrier. The blood-brain barrier is a physiological barrier that keeps many substances that circulate in the rest of the body from getting into the central nervous system, thus restricting what can cross from circulating blood into the central nervous system. Nutrient molecules such as glucose or amino acids can pass through the blood-brain barrier, but other molecules cannot. This actually causes problems with drug delivery to the central nervous system. Pharmaceutical companies are challenged to design drugs that can cross the blood-brain barrier as well as have an effect on the central nervous system. Oligodendrocyte is the glial cell type that insulates axon in the central nervous system. There are a few processes that extend from the cell body. Each one reaches out and surrounds an axon to insulate it in myelin. One oligodendrocyte will provide the myelin for multiple axon segments, either for the same axon or for separate axons. Microglia are smaller than most of the other glial cells. Their function is related to what macrophages do in the rest of the body. When macrophages encounter diseased or damaged cells in the rest of the body, they ingest and digest those cells or the pathogens that cause disease. Microglia are the cells in the central nervous system that can do this in normal, healthy tissue, and they are therefore also referred to as CNS resident macrophages. The ependymal cell is a glial cell that filters blood to make cerebrospinal fluid, the fluid that circulates through the central nervous system. Ependymal cells line each ventricle, one of the four central cavities that are remnants of the hollow center of the embryonic neural tube. The choroid plexus is a specialized structure in the ventricles where ependymal cells come in contact with blood vessels to produce cerebrospinal fluid. Because of this, ependymal cells can be considered a component of the blood-brain barrier or a place where the blood-brain barrier breaks down. They also have cilia on their apical surface to help move the cerebrospinal fluid through the ventricular space. Satellite cells are found in sensory and autonomic ganglia, where they surround the cell bodies of neurons. They provide support, performing similar functions in the periphery as astrocytes do in the central nervous system, except for establishing the blood-brain barrier. The Schwann cell insulate axons with myelin in the periphery. Schwann cells are different than oligodendrocytes in that a Schwann cell wraps around a portion of only one axon segment and no other. Oligodendrocytes have processes that reach out to multiple axon segments, whereas the entire Schwann cell surrounds just one axon segment. The nucleus and cytoplasm of the Schwann cell are on the edge of the myelin sheath. Myelin is a lipid-rich sheath that surrounds the axon and by doing so creates a myelin sheath that facilitates the transmission of electrical signals along the axon. It also includes important proteins that help to hold the layers of the glial cell's membrane close together. The myelin for axons is provided by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. For oligodendrocytes, the rest of the cell is separate from the myelin sheath as a cell process extends back towards the cell body. A few other processes provide the same insulation for other axon segments in the area. For Schwann cells, the outermost layer of the cell membrane contains cytoplasm and the nucleus of the cell as a bulge on the one side of the myelin sheath. To form the myelin sheath, the glial cell is wrapped around the axon several times with little to no cytoplasm between the glial cell's layers. In cross section, the myelinated axon appears as a nearly circular profile surrounded by a multilamellar spiral sheath. Amazingly, a large myelinated axon may have up to 250 to 300 turns of myelin wrapping around it. During development, the glial cell is loosely or incompletely wrapped around the axon. The edges of this loose enclosure extend toward each other, and one end tucks under the other. The inner edge wraps around the axon, creating several layers, and the other edge closes around the outside so that the axon is completely enclosed. An unmyelinated axon does not have a myelin sheath because it is only covered by a single cell of glial cell cytoplasm. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.